So as Frank said, I'm Aaron. I'm co-founder, CEO of Reservoir Neuroscience. Today I'm going to tell you about how loss of health in your blood vessels, which we all experience, drives brain aging. But before I do that, I just want to ground us a little bit in how we think about aging. And to do that, I want to come to this really interesting photography project by a photographer, Jan Langer, called Faces of Century. So these are photographs of people when they're young and then when they reach 100 years old. So it's a group of centenarians. And I just want you to look at these with me and kind of just take a moment to really reflect on aging, at the personal level. And I'll flip through them and just let the images hit you and think about what you feel like you're seeing about aging and how that reflects on what you know about aging intuitively. And there's more of these if you want to look up the photographer, but I think I see different things actually every time I look through these, but what I'd like to focus on today is the idea that aging is not just one thing. If we look at these people, we see that they are all very individualistic in how they aged. There's not one outcome. I think you can almost literally see their life experiences being kind of written in their body, and all those experiences were different for the different people. That idea is very powerful, but it's almost completely opposite to how we approach the science of brain aging. So when we think about brain aging, I think almost all of us, and the field in general, thinks about Alzheimer's disease. And when we think about Alzheimer's disease, we really just define that by one feature of disease. So instead of this individualized aspect of complex biology, we say, well, Alzheimer's is amyloid beta. And if you get amyloid beta in your brain, you have Alzheimer's, and then you'll get dementia. And so what we've tried to do at Reservoir Neuroscience is go back to maybe this more biological approach, right? How do we think about true biological aging and then how that drives brain aging and brain disease, specifically dementia? And to sort of get an inroad into how I think about that, I just want to share two very simple pieces of data with you, but I think that are extremely powerful to help us shift our perspective around brain aging. The first is that if we really try to understand and track people and understand why people are getting Alzheimer's, what I'm really convinced by at this point is that Alzheimer's is a lifestyle disease. So forget amyloid beta, forget genetics. For most people, for most of us in this room, the reasons that we'll get Alzheimer's disease are most linked to lifestyle factors. And you can see some of these largest factors up here on the screen. This really defines for almost everyone the largest patient segments why we get Alzheimer's disease and what we can actually attribute into early life risk leading into later dementia and Alzheimer's. And the second piece of data that I want to share with you is that this looks like it may be either preventable or at least something that we can slow and really adjust through our lifestyles. And so just one very, again, simple piece of evidence showing that is that people who exercise as compared to the general population have this huge risk reduction in Alzheimer's, right? So this is powerful for several reasons. Firstly, this risk reduction is not linked to even the brain in any obvious way, right? It's not linked to amyloid beta or to the things we typically think about with brain aging. It's a change in the lifestyle. But what's even more important is that if we look at the magnitude of that risk reduction, it's extremely powerful. Of course, we can't directly compare epidemiological data to clinical data, but we know that the latest anti-amyloid beta antibodies, which have just passed through their clinical trials, lecanemab, they give at best maybe like a 6-month delay in the onset of the disease, right? So you might slow progression of your disease by 6 months if you take these amyloid beta reducing antibodies, which is not terribly exciting to me. At the same time, if we look at a lifestyle intervention, we see this huge effect in improving disease outcomes. And so what I'd really push us to sort of challenge us to do is to think about, can we understand the biology of this lifestyle, and can we understand some of the ways we might target that biology and replicate these kinds of much larger impacts in improving disease outcomes, hopefully at a drug level. And we're very fortunate now because probably for the first time in history, we now have the data to be able to do that. So the Alzheimer's Disease Neuroimaging Initiative is the largest patient data set ever for Alzheimer's disease, and it's been tracking thousands of patients now longitudinally over many years, gathering pretty much every different type of biomarker that the investigators are able to gather and know how to gather. And so for this first time, it really allows us to look at all the factors of Alzheimer's disease and try to understand what's really driving patient outcomes. And what's extremely provocative about this is that the data is now in, and we can see and trace which types of factors show the biggest effect size in showing abnormalities and being correlated with patient outcomes. And I just want to highlight a couple things. I know there's a lot of stuff on here, but the amyloid beta, which is a traditional marker of Alzheimer's disease shown here in red, the deposition of amyloid beta is not the earliest appearing factor or the largest factor in this unbiased, largest ever clinical data set. What we see is that for most people, the first sign of brain aging that you can really detect is loss of vascular health. And that vascular health appears before amyloid beta buildup, but it also has a larger abnormality. So the signal to noise, the ability to predict patient outcomes is greater for vascular pathology. And I can't really overstate how important this is. In my mind, if you go back decades, this red line has always been the textbook. If you learn anything about Alzheimer's disease, this is what you learn about disease progression. And what we now know is that it's probably not the most important feature of disease, if you believe unbiased large data sets. So what can we do about it? We can look into patients' brains and follow these patients, and we can find the patients who have vascular pathology, and we know through efforts like the Alzheimer's Disease Neuroimaging Initiative that this vascular patient subset actually defines the largest patient subset. So most people have this vascular pathology, and again, it's showing the largest effect of driving disease outcomes. Because of that, we launched Reservoir Neuroscience with the goal to develop the first drugs ever that are intended to target and reverse vascular pathology. We believe that if you can heal the vascular outcomes, that's going to give us the largest effect of reversing the brain outcomes and recovering and rejuvenating brain function in Alzheimer's. And when we really dig into this biology, to me, it's much more intuitive than the amyloid beta hypothesis, and there's a very clear connection between the lifestyle and how it drives disease. And we can break that down into pretty simple factors. Essentially, as you age, all of us start to accrue loss of blood vessel health. This is primarily driven by lifestyle, high blood pressure and atherosclerosis, aging of our immune cells, ApoE4, which is the largest genetic risk factor for Alzheimer's disease. All of these different lifestyle factors cause wear and tear on our blood vessels, various types of damage that accumulate over aging. That wear and tear on the blood vessels converts the blood vessels from a healthy tissue into essentially a diseased tissue. And this diseased tissue then becomes activated and highly inflammatory. Activation of that highly inflammatory process causes then the disease to spread from blood vessels into the brain and to trigger all sorts of disease-causing and inflammatory processes in the brain. And so we believe that if we can reverse some of that, we can essentially cut off the largest source of disease-causing chronic, unrestrained inflammation in the aging brain and help it to recover to a healthy state. We've done that really in two phases now in the company. There's the historical phase where we did our drug discovery and development, so I'll tell you a little bit about how we approach that. And then there's the future phase, which is how we want to bring this drug into human clinical trials and why we think it will work. From the drug discovery side, we started really, we think of the vasculature as really an undiscovered country. There hasn't been any drug development ever for trying to reverse this kind of vascular pathology. So we wanted to start from a place where we could try to develop really try to find the strongest possible intervention. What's going to have the biggest bang for our buck? What's going to have the biggest impact on improving vascular function? To do that, we developed an organ-on-chip technology. So we grow human blood vessels inside of the lab. This is all human cells. It realistically replicates the complex three-dimensional architecture of your blood vessel. It's real flow through it. It essentially looks and acts as close to the blood vessels in your brain as what we can do scientifically. That allows us to then directly test interventions, flow drugs through diseased blood vessels, and look at what has the biggest effect in actually recovering health and function in essentially a real blood vessel. That discovery process and that technology led us to screen many different types of targets. I'm showing you some examples here. And what we found in particular is that there was one target that really popped out, that when we antagonize this target, we completely restore vascular health. This is the largest effect size that we've ever seen in our discovery platform. And we can bring those blood vessels all the way back to a healthy state where they're now completely functional and no longer show any of the markers of pathology that we follow. This target is the prostaglandin EP2 receptor. And as we understand more and more about the biology, it really starts to make sense why it plays a central role in disease and how the mechanism works. What's really critical to understand about this target, this receptor, is that the receptor itself, which is shown here, is not present on healthy blood vessels. And so if you go into a young person or a healthy person, you won't really see this receptor at high level on the brain's blood vessels. But what happens is that it actually becomes upregulated in response to these other insults and triggers and types of damage to the blood vessels. So this particular receptor is like a first responder. When your blood vessels start to accrue damage, it becomes upregulated, and then it's a master regulator that turns on inflammation and puts those blood vessels into a state where they essentially have chronic, unresolved inflammation. It's really important that once this receptor becomes activated, it stays upregulated and active over long periods of time, and so it really matches what we see in the brains of actual Alzheimer's patients, where you have inflammation that basically never stops. And it's pathology that continues to accrue over a chronic period of time. And there's been, fortunately, a lot of great biological characterization of this receptor, and we now know that in over 5 models of animal models where there's vascular pathology, every time that someone tries to antagonize this target, we improve vascular outcomes for mice. And what's really important to us there is that biological concordance, right? So sometimes there's this problem in discovery of biology. You have one animal model, you get your hit, it seems like it works, but maybe that model doesn't replicate the biology. If the biology really has the impact that we think it should, we would like to see that in every model we look at, or at least in most of the models we look at, antagonizing this target therapeutically actually delivers the outcomes that we want. And that biological concordance is exactly what we see with the prostaglandin EP2 receptor. In particular, I'd just like to highlight one very exciting study that was done out of a fantastic lab in Stanford, where they found that antagonizing this target with 2 different small molecule antagonists completely reversed neuroinflammation in aged mice back to a youthful level. And then after reversing that neuroinflammation, the cognitive function of the mice was completely recovered. So we think the biology is extremely powerful and captivating. And after we did our drug discovery, we then launched a program to develop our own proprietary small molecule antagonists. Essentially, that's just a traditional SAR medicinal chemistry program. But what's the most important take home is what we've done through this program is develop what we feel are the class-leading EP2 antagonists, the best EP2 antagonists that have ever been made. These are biological, clinically relevant drug-like molecules that have orders of magnitude improvement in the clinical properties compared to previous generation compounds. So they're orally bioavailable, they're potent, they're more potent than any compound that's ever been made, and they're very metabolically stable and suitable for once per day oral dosing, ultimately, hopefully in a human patient. Now that we've developed this compound, I'd like to tell you a little bit about what we think we can do with it, and there's 2 aspects that are particularly exciting. First, because we're able to measure this vascular pathology with a simple MRI, so a 45-minute session, we can measure multiple domains of vascular pathology. we now have a very precise and personalized way to identify the actual biological clinical patient population that we want to treat. And so we can go into a patient population who's broadly diagnosed with Alzheimer's, find the majority of those patients who, like I said, most of us have vascular pathology underlying that. We can identify those patients that have vascular pathology, and when we do, we know from large-scale clinical data sets that those patients actually, as we expect, have worse outcomes. So the patients with the vascular pathology have faster cognitive decline, they're at a higher risk, but equally importantly, they're a rarely nuanced select patient population within the larger patient population. And so it gives us a biomarker-based approach to run a human clinical trial where we give the right drug to the right patient. And even more exciting, in the first phase of the clinical trial, we can potentially measure early biomarker outcomes and see if we're delivering measurable improvements in human vascular biology even before we measure cognitive outcomes in phase 2 and phase 3 clinical trials. The other thing that's very exciting is that we have some real-world data showing that this therapy might work. So you may not know, but probably everyone in this room has already taken a drug that indirectly inhibits or suppresses EP2. And that's because EP2 is downstream of NSAIDs. And so NSAIDs, like ibuprofen, which we've all taken, they have very non-selective. They hit a lot of different things relating to prostaglandin pathways and pain management. But downstream of NSAIDs, we do see a reduction in EP2 as one of the primary effects of NSAID usage. And because we're all taking them, we can actually look at epidemiological data, and this has been done in many studies over the years, going back decades. And we know that patients who have taken chronic NSAIDs, like ibuprofen, for up to 2 years have the largest effect size in reduction of Alzheimer's that I've ever seen for any intervention. So in the largest study, we saw a reduction of 80%, so 80% less incidence of Alzheimer's in patients who are taking ibuprofen. And so we think that this is incredibly powerful, going back to the biology. If we can drug this target, but in a more precise and sophisticated way, we may be able to create these kinds of extremely dramatic improvements in actual real-world Alzheimer's patients who have vascular pathology. Now, I think I know this audience, and I'm a little bit worried that there are probably quite a few of the 40 supplements a day type here in the audience. So probably some of you are now wanting to run out and start to take as much ibuprofen as possible. Before you do that, I'd like to advise you that chronic use of NSAIDs like ibuprofen is not safe. So one of the problems with NSAIDs is that they really do a lot. They work on this COX-2 enzyme, and they have a lot of downstream targets that they hit. So we call them selective in a sense, but they really aren't selective. And in particular, we know that chronic use of NSAIDs can lead to some really extremely important and serious side effects, cardiac failure and gastric bleeding, ulcers that can be lethal. So while it does show us the power of this potential therapy, I wouldn't advise that we start to pursue this as a therapeutic approach. But again, this helps us understand what we really want to do with our target, which is EP2. EP2 is very selective within this pathway. It has no known side effects. It's been dosed at chronic levels in mouse. There's no safety concerns whatsoever. And it's really selective for, in particular, the chronic inflammatory arm of the pathway. So as I said before, the receptor becomes upregulated and really helps to create chronic inflammation within the target cells that never gets resolved unless you potentially have a drug intervention. And so we think that this is a selective way to kind of take a well-known biology, but really go after it in a personalized way where we apply it to the problem of vascular pathology and have a potential for a safe, efficacious improvement in patient outcomes. So I would just like to also thank the wonderful team that we've worked with, my co-founder Vlad, who's somewhere here in the audience, kind of been the scientific driver in many ways of the company and been a huge privilege to work with him, and some of our wonderful investors and board who I think are really synergistic with our vision and helped us so much to drive the company forward. So with that, I'll take any questions. Thank you.
Yeah, let's do that. So.